All right, we're going to pick up in the last video with the accessory organs. So we've covered the alimentary canal, so now we're going to jump into our special accessory organs. The first one we'll look at is the pancreas. The pancreas you studied during the endocrine system. It has both endocrine and exocrine functions. As an endocrine organ, you probably remember, hopefully remember studying the hormones insulin and glucagon. And that is, of course, going to affect your blood sugar. Now, as far as the digestive system, it's mainly an exocrine secretion. And this means it's going to release enzymes that are really, really important in chemical digestion in the small intestines. Now, the enzymes are released into the ducts of the pancreas that empty into the alimentary canal. There is a duct, the main pancreatic duct, that travels down the middle of the pancreas. This is where it receives all of the secretions that the acinar cells are going to make. Now, right near the duodenum, the main pancreatic duct merges with the duct from the liver and gallbladder, and it's going to empty into the duodenum. There's also a smaller pancreatic duct that's used, but this main pancreatic duct is the main pathway. Now, pancreatic juice is really, really, really important for chemical digestion. It's a collection of different things from the duct cells and the acinar cells, a lot of water, multiple different types of enzymes, and of course proteins. The duct cells also secrete bicarbonate, which you already know is a base that's used to buffer the really acidic kind that comes from the stomach. It also helps protect the duodenum from damage by that acid, because remember, the small intestines is not built to withstand a really low pH. The digestive enzymes are crucial in chemical digestion. They help catalyze the breakdown of carbs, proteins, lipids, and nucleic acids. Now, pancreatic secretion does occur at a basal rate in between meals. Now, during eating, obviously, that secretion rate is going to rise because of different hormone stimulation. All right, so that gets us to the liver and gallbladder. The pyramid-shaped liver is in the upper right quadrant. And right underneath it, a really small sac is present called the gallbladder. Now, these two organs have close relationship. This is getting into some anatomy of the liver, which is lab. You can see it's covered by the visceral peritoneum. It has 4 lobes. Your right and left are the easiest to locate it. There's a ligament, the falciform ligament, that's going to separate the two. It also anchors it to the abdominal wall. You have the round ligament, which is on the inferior surface. It's actually part of your umbilical cord that was present as a fetus. You have your 2 smaller lobes, the caudate and quadrate, and they're in the back of the liver's right lobe. And then you have a port, and we call it the porta hepatis. And this is where numerous blood vessels enter and exit the liver. The hepatic artery, the hepatic veins, and your hepatic portal vein. Nerves, lymphatic vessels, and the common hepatic duct also enter and exit through this port. So this is like the grand central station. You can see it in this image right here. You can see your lymphatic vessels coming up, arteries and veins all come through that porta hepatis. Now, this is the histology picture of a liver lobule, and this is the basic unit of the liver. The liver cells that make up this are called the hepatocytes, and they actually have kind of like a hexagonal shape, and they're stacked on top of one another. You also have a portal triad. Now, I'm going through this quickly in the videos because this is really lab stuff, all right? And you can see this in your book as well as your lab book. But this portal triad has each of the 6 corners of the lobe or the lobule. And you're going to have your hepatic arteriole, your portal venule, and a small bile duct, of course, that carries the bile. Now, the hepatic arterioles and the portal venules both drain into these large leaky capillaries known as your hepatic sinusoids. The sinusoids have walls. They're not continuous. This, of course, is made so that big stuff can get in and out of the bloodstream. Now, blood flows through these sinusoids, and the materials are exchanged between the blood and the hepatocytes, and eventually they're all going to drain into the central vein. And the central vein merges and drains into the hepatic veins, which then are going to go back up into the inferior vena cava. So again, that's just returning that blood back to the heart. Bile flows through the liver lobe in the opposite direction. It goes from the hepatocytes into the tiny ducts, which are called the bile caniculi, and then eventually it drains into your bile duct. All right, so what is the functions of the liver? Your liver has one of the most diverse sets of functions compared to any other organ in the body. It has endocrine and exocrine secretions. It also is going to convert bad toxic chemicals into non-toxic substances so that the body can get rid of them. The main function, digestive-wise, is to produce bile. And bile has multiple things, a lot of water, electrolytes, some organic compounds, and there's two big functions for bile, required for digestion and absorption of lipids. Remember, lipids cannot be broken down as they are in the small intestines. They do have to be broken down. That's done by emulsification, by bile. And then you also have the mechanism by which the liver excretes waste and stuff that your kidney cannot get rid of. Liver produces the bile on a continual basis, but it doesn't secrete it at a basal rate. So it's produced continually, but it doesn't get secreted unless it's stimulated to secrete. One of the main organic compounds in bile is the bile salts. This comes from cholesterol. Bile salts are amphiphilic, which means that they have both polar and non-polar regions. So this is important because it allows them to interact with the watery environment of the small intestine as well as the lipids in the small intestine. And it basically coats the lipids in the duodenum, and it physically breaks them into smaller pieces, and this is called emulsification. This is a type of mechanical digestion which is absolutely necessary in order to chemically digest the fats and of course absorb them once they're digested. Bile has some other things that the liver excretes: cholesterol, waste products, and of course some toxins. And this is how your body rids of those. All right, some other liver functions. It excretes bilirubin, which is a waste product that comes from the spleen when the spleen breaks down hemoglobin. You recall hemoglobin is a protein in the blood, and the spleen is the graveyard for red blood cells. So hemoglobin is present in those red blood cells, and when it's broken down, you have a product of bilirubin. Some stays in the blood, others sent to the kidneys, and this is actually what gives your urine its yellow color, is this bilirubin. There are some other things that it does. Nutrient metabolism, going to get nutrients from the diet, detoxification, produced by the body, as well as things that we eat and drink. We talked about alcohol during class. This is how we turn that alcohol into a non-toxic substance. And then excretion. It directly gets rid of bilirubin in the bile, along with some other things that the liver processes that need to get out. It also has particular certain antibiotics, and it modifies things so that they can be excreted by the kidneys. Okay, so that gets to the gallbladder, small little tiny sac that's on the bottom, the back of the liver. It receives most of its bile from the common hepatic duct, but it's going to store, concentrate the bile, and it releases it when it's supposed to. That stimulation of release is regulated by a hormone called CCK. And basically the smooth muscle in the gallbladder wall contracts, and that releases bile into the cystic duct. Now the cystic duct joins the common hepatic duct, and that becomes the common bile duct. The common bile duct joins the main pancreatic duct near the duodenum, and it forms the ampulla. This is the hepatopancreatic ampulla. That ampulla is surrounded by muscle, smooth muscle, and it forms the hepatopancreatic sphincter. This controls emptying bile and fluids into the duodenum. The main pancreatic duct is going to bring all of the pancreatic juices into the small intestines, and then the common bile duct is going to bring all the bile there. The bile has salts and cholesterol, and sometimes they can form hard lumps called gallstones. You've probably heard of those before. Okay, so this is another chart, just like I showed you for your alimentary canal, but this is for accessory digestive organs. And you have your organ, the structure, and the function. Okay, so let's kind of look at an overview. The digestion, we know, breaks down food into smaller pieces. There's two types, mechanical and chemical. We've already talked about mechanical is physically breaking it into smaller pieces, mastication in the mouth, churning in the stomach, segmentation in the small intestine, and then you've got chemical, which is breaking the chemical bonds between the food. This varies for each nutrient because there's specific digestive enzymes that they're going to target specific nutrients. Most enzymes catalyze hydrolysis reactions, and that means, you learned this back in 103, that means you use a water molecule to break two things apart. And this explains why so much water is secreted in the fluids. Your gastric juices and your pancreatic juices, water is a key component. The digestive enzymes that speed up this are called the enzymatic hydrolysis. And then, of course, once they're digested, then they have to be absorbed and enter the body. Now, let's look at carbohydrates. This digestion begins in the mouth with salivary amylase, and it's going to break long polysaccharides into shorter oligosaccharides. Generally, there's very little actual digestion that takes place in the mouth just because food's not in there long enough. But it does continue down into the stomach, but the stomach acid usually inactivates that salivary amylase before it actually completes that task. Now, the chemical digestion of carbs resumes when they move into the small intestine, and this is when they come in contact with pancreatic amylase. Pancreatic amylase is going to come from the pancreas. It's one of those really important enzymes in pancreatic juice. Notice amylase. When you see the term amylase, you know we're breaking down carbs. Salivary amylase means it comes from the saliva in the mouth. Pancreatic amylase means it comes from the pancreas, but it's going to act in the small intestines. The result of activity of all of those is going to release smaller carbs, monosaccharides, glucose, fructose, and then, of course, once they're in the blood, then they're delivered to the liver by the hepatic portal vein for processing. Excuse me. Now we're going to move to proteins. Chemical digestions. And protein doesn't start until the stomach when they get in or they encounter pepsin. Now, if you recall, a review, this material is a review from when we studied the stomach. Chief cells are going to produce pepsinogen, which is an inactive precursor for the enzyme. When pepsinogen comes in contact with low pH, it's going to be activated and become pepsin. Activated pepsin is actually what digests the proteins. And this really only catalyzes about 15% of the proteins in the food. The majority of protein digestion is in the small intestines with the help of trypsin. Now, the pancreatic enzymes, one of the main ones that's along with pancreatic amylase is trypsinogen, and it is the precursor to trypsin. Trypsinogen, when it comes in contact with the brush border has all those villi and microvilli that really bring it into contact with a lot of chemicals. When it comes in contact with the brush border, trypsinogen turns into trypsin, and trypsin is the activated enzyme that digests proteins. Most protein digestion is going to take place here in the small intestines in this particular day. All right. Just like carbs, amino acids, they're put into the blood and they're delivered to the liver through that hepatic portal vein, and of course that's processing. Okay, let's look at lipids. The majority of lipids are taken in your diet by triglycerides, and they tend to stick together. And when they stick together, they form these large globules instead of being evenly distributed in the solution. So we have to break up those lipid globules. The first one is by mechanical digestion. Once again, mastication in the mouth, churning in the stomach, and segmentation in your small intestines. And then, of course, our bile salts are going to emulsify the lipids, and we discussed that. This gives the enzymes enough surface area to really work and efficiently digest those lipids. And so this is just a visual of kind of seeing how this works. And then another one to show that absorption of lipids. All right, lipids are different from carbs and proteins because they're not delivered directly to the liver after absorption. The hepatic portal vein delivers leftover bile salts to the liver where they make new bile. The lipids travel within chylomicrons through the lymphatic vessels and eventually to the thoracic duct where they join the rest of the lymph. Now, as they travel through the blood capillaries, these chylomicrons, the lipids are progressively removed and enter the cells where they can be used for many different purposes. But carbs and proteins go back to the blood. Lipids are going to go to the lymphatic system. Remember, we talked about that when we learned lymphatic system, that this is the one thing that lymphatics is going to have to really process instead of the blood. All right, so here's your summary of carbs, proteins, and lipids, okay? And this kind of shows who's acting where they're going to be returned to, and it also gives you the enzymes that are going to do this digestion. Okay, last one's nucleic acids. Some of the foods we eat have nucleic acids in them, and we have to use enzymes called nucleases. They're going to break nucleic acids into their nucleotides. This also happens with the brush border enzymes. They remove the phosphates and the sugars, and then these are absorbed through the primary and secondary active transport mechanisms from the capillaries into the villi. Your alimentary canal, this is just kind of looking at water and electrolytes. Your alimentary canal absorbs a lot of water, a lot of electrolytes, and a lot of vitamins. Most of this occurs in the small intestines, but a significant amount does happen in the large intestines. This is really interesting to look at these numbers. On average, your small intestines receive about 9 liters of water. 2 liters of that is ingested, like from your food and drink, and then the remaining 7 is secreted by the alimentary canal itself, along with the accessory organs. Of this 9, about 8 liters are absorbed into the enterocytes of the small intestines. Most of the remaining water is going to be absorbed by the enterocytes of the large intestine, and that leaves only about 0.1 liters is actually excreted in the feces. So this goes to show you as the absorption content that the intestines have to do in order to achieve water homeostasis. Okay, last thing is your vitamins. These are chemicals that are provided by the diet, and they're really important in a lot of metabolic reactions. You have 2 kinds, water-soluble and fat-soluble. Water are polar, that means they have to be absorbed in the small intestines with water, and then fat-soluble is they're lipid-based, so they're nonpolar, and they're packaged into micelles in the fats, and they have to be absorbed along with food and fats. All right, and then there's your big picture of digestion. Again, most students think that this system makes a lot of sense. You're more familiar with it, and it tends to come a little bit easier. So play these videos as many times as you need. Really, really important to use these images from your book that I've incorporated into the PowerPoints. I put them there for a reason, so please make sure that you do look at these images.